Well, good morning, everyone, and welcome to the Insects and Pests Survey of 2024 webinar. My name is Quinn Cubben, and I am the Crop Extension Specialist with the Ministry of Agriculture in North Battleford, and I will be today's chair. CEUs are being offered for CCAs on this webinar. In order to get credit, you need to attend the entire live event, and there will be a QR code at the end of the presentation to collect credit. If you missed the code and you have attended live, please email us afterwards and we can get everything sorted out for you. Before we start the formal presentations, I have a few items to share. Our crop extension specialists put on this webinar series each year. We work out of 10 different offices across the province, and as you can see here on the map, each office contains a wide variety of specialists that can help you with any agricultural-based questions. We have offices in North Battleford, Prince Albert, Tisdale, Cannersley, Outlook, Humboldt, Yorkton, Swift Current, Moose Jaw, and Weyburn. If you have ever heard the words on the radio, Saskatchewan is 98% complete harvest this week, then you have likely heard of the crop report. Our colleagues out of Moose Jaw office write the crop report each week during the growing season based on data collected from crop reporters across the province. We are always looking for new crop reporters, so please use the QR code to sign up today. Crop reporters are a mix of producers, agrologists, and everyone else interested in agriculture. They take about 5 minutes to fill out, and you fill up for the surrounding crop districts. I also wanted to mention the 2024 Crop Diagnostics School. We will be in person this year, and it will be held on July 24th and 25th in Melfort. More details to follow the event, along with other upcoming field days hosted by local agri-arm research sites. Topics will include insects, weeds, diseases, soils, and herbicides. Lastly, we wanted to make sure everyone is aware of the funding available through the Sustainable Canadian Agriculture Partnership. We offer a wide variety of programs that can help meet your needs on your operation. Please reach out to the local agriculture program specialist to find out any of the funding you may qualify for. Along with this information, we also have additional resources for everyone under our handout section in the chat box at the toolbar. This PowerPoint, a document listing valuable resources to be aware of, and information on upcoming events and opportunities are all located there. They will be shared along with the recording after the webinar is complete. Today's presentations will be followed with a live Q&A with Alireza and James Tansey. The main hub for today will be your webinar screen. As mentioned, we will share relevant downloadable handouts with you in the chat box area. The entire session will be recorded, so if you miss anything and wish to revisit any of the presentations, you will be able to. Recording links will be sent out later today in a follow-up email. An evaluation will pop up at the end of the webinar this morning. Please ensure that you fill out this evaluation, as this helps us determine what topics you want to learn about and helps us prioritize research needs for the future. We have a few housekeeping notes to keep in mind during the webinar. If anybody has any questions during the presentation, and you may want to type them in the chat box at the bottom right hand of your control panel. We will try to get as many of your questions as possible, but may need to limit them due to time. Your questions will remain anonymous, and we will only be sent out to me. During the presentation, you will be muted so that you can ensure there's no feedback that will interfere with the audio. We will start with the day off with Dr. James Tansey, who has been the provincial entomologist with the Ministry of Agriculture since January 2018. Prior to this, he worked as a research scientist with ALK in Post Falls, Idaho, investigating and optimizing industrial production and allergen expression in in-house dust mites. He came to Idaho from postdoctoral fellowship at the University of Florida, where he investigated control in the Asian psyllid and other citrus pests. His doctoral research and his postdoctoral on canola entomology in the Dostal Lab at the University of Alberta primarily hosts plant resistance to some major insects of canola in novel germplasms. His master's degree examined insect-plant interactions in an important leafy spurge biological control agent. We will start whenever you are ready, James.
Okay, can you hear me now?
You're a little bit fuzzy, James.
That just might be my personality.
Still sounds a little bit staticky.
Okay, I'm going to, I'm using a headset right now. I'm going to unplug the headset and see how the microphone works on my computer for this. So, yeah, how's that?
Yeah, it's a little bit better.
Okay, let's go with that then. Okay, trying to show my screen right now. Do you see my screen?
Yes, we can.
Okay, I'm going to. You see full screen?
Nope, we just see the PowerPoint.
Okay.
James, I can share it through mine. Here.
Yeah, why not? You're probably more time efficient. So. Excellent. There it is. There's one small change that I made to the version that you have, and I'll illustrate that when we get to it. If you could go to the slide, please. Okay, lots of questions about grasshoppers this year. Uncertain. A lot of pressure last year, at least local pressure in several regions. Pardon me, this is maybe this thing may be very strange. Okay. Which you probably can't see, but there it is. Okay, I'm back to normal. So looking at the 22 grasshopper survey, of course, we have very dry conditions in 2021. We saw an upregulation of populations in 2022, particularly in those south central regions. 2023, of course, another dry year. 2022, some dry conditions, hot conditions as well. And so we've seen a year-over-year increase in grasshopper populations. Culminating in some pretty significant populations in 2023. Go to the next slide, please. When we're talking about grasshopper populations, we're dealing primarily with 4 species, and this was demonstrated in the surveys as well. So, the most important pest grasshopper that we're seeing currently is the two-stripe grasshopper. All of the pest species that we're talking about overwinter as eggs. So, a lot of the factors that contribute to population increase or decrease are going to be influential on all 4 of these species. So once again, all 4 overwinter as eggs. In the case of these ones, the eggs start to hatch about 8 to 10 days ahead of one of the other major pest species, migratory grasshopper. And there will be some succession in the emergence in the hatch of these animals, but a lot of conditions, as I mentioned before, are going to be influential on them. So eggs are typically going to be concentrated on roadsides and ditches for this one. They're really like cracks in the soil, and some actually, like the grasshopper, will actually seek out dense thatch for lower position. The two-stripe, however, do prefer lush habitats and heavier textured soils. They tend to prefer lush foliage. But they will feed on cereals and can be significant pests in cereals. Although cereals are not their favorite meal or their favorite host plant. You can see also that their range extends over the bulk of North America, even northern Mexico. And they can be migratory in parts of this range, including in this part of the range. And certainly saw large numbers of them taking flight this past summer. So 5 nymphal instars. That 4th instar, the bright green one, is typically where the grower is going to spot numbers of these. They're very conspicuous, being bright green, of course, and you can see that characteristic black stripe on the hind femur. Good way to tell them apart from other species. In addition, when you're looking at the adults, that the two stripes extending from between the eye to the tips of the wings. These girls are big as well, so the females can be about 1.5 inches or larger. Females are larger than males. And if we go to the next slide, please. Which is the case with grasshoppers in general, is that the females tend to be larger than males. Another important pest species in the prairies is the migratory grasshopper. And most prior outbreaks, essentially all prior outbreaks, historically have been associated with this grasshopper species. The current outbreak seems to be dominated by two-stripe, or is dominated by two-stripe, and by no small measure. So exactly why this species is becoming dominant rather than migratory grasshopper, which seems to be relatively restricted to southern regions, although they can be quite numerous, is a bit of a head scratcher at this point. So once again, overwinters eggs, the hatch is going to start in May. And of course, we had the hatch beginning in late April for two-stripe grasshopper in some parts of the province this past year, all depending on heat accumulation. We had a very warm May. So in the case of migratory, the hatching is going to start along open south slopes and areas with little vegetative cover and sandy soil. So that is areas with good heat accumulation in the soils. Once again, five nymphal instars, really characteristic. So can you hear me, Alec?
Yeah, we're just having significant issues with your audio. I think what we're gonna do, folks, is we'll get James to phone in through a phone call, but maybe if you're okay with it, we'll switch to Alireza doing the speaking first, and then I can work with you in the background, James, to get clearer audio.
Yeah, no, that's if you're not hearing me, there's not much point.
Yeah. Okay, so we will switch it over to Alireza. Alireza, can you hear us here?
Yes, I can start, and then you can figure out what.
Perfect. So
what the technicals should be for James.
Awesome. We will get Quinn to run through your intro, and I will help James in the background here.
Yeah, thanks, Ali.
So Quinn, can you confirm that
Yeah.
my presentation can be seen and it is on the presenter mode?
Yeah. Okay, so next up here we have Dr. Alireza Akhavan. Alireza is the Provincial Specialist Plant Disease with the Saskatchewan Ministry of Agriculture. Since August 2020, prior to joining the ministry, he has been with the University of Alberta and Agriculture and Agri-Food Canada in Macomb for almost 10 years, teaching plant pathology and working on various diseases of canola and cereal crops across the prairies. We'll start whenever you're ready, Alireza.
Sure. So good morning, everyone. Without further ado, I am going to share some of the findings from the 2023 Saskatchewan Crop Disease Survey programs. We will have a longer portion on canola because canola, in addition to the general canola disease survey, it has two separate monitoring programs applicable to it, which is collaborative monitoring program and verticillium stripe monitoring program. So before I start, I just wanted to thank all the contributors to this work. I am just speaking on behalf of a large group, including provincial specialists, crops extension specialists, and crops protection laboratory from Saskatchewan Ministry of Ag, Farm Plant Health Technical Advisors of Saskatchewan Association of Rural Municipalities, Saskanola Discovery Seed Lab, University of Manitoba, and University of Alberta. Both help us in some of the specific testing. And we had the honor of being helped by industry surveyors from multiple organizations like Meadow Lake Co-op, Bayer, Nutrient, Canola Council of Canada, and some volunteer agrologists and producers. My colleague Carter Prue, our integrated pest management specialist, has a big role in coordinating all the surveys, while our other colleague Raul Avila, who is the pest regulatory provincial specialist, has the program of collaborative monitoring under his portfolio, as chloride is still a declared pest in the province. Also, we had the great help from Corey Jacob and Dale Risolo, our old seed specialist of the time, at the time that the surveys were being done, and Dale Risolo is our pulse and specialty crop specialist. So, again, when it comes to canola, there are 3 separate programs that monitor diseases of canola. One is General Canola Disease Survey. This year we had 217 fields surveyed under that program, but we also have 2 separate programs in addition to General Canola Disease Survey. One is Verticillium specific survey, which is done after harvest, usually during the month of October, so it's a fall survey. And this year we surveyed 102 fields at that time for Verticillium stripe monitoring. We also have a comprehensive program for clubroot, perhaps one of the most comprehensive program for clubroots internationally. And we looked at 534 fields. We examined them for the presence of clubroot. So for the General Canola Disease Survey this year, 217 canola fields were surveyed, and survey completed between August 1st and September 27th. 20 plants is getting examined from each 5 sites along a W pattern in each field. So for each field, 100 plants in total gets examined for all the diseases of canola. And during this survey, incidence and prevalence was recorded for diseases, and severity was rated for some diseases. Just to refresh minds, prevalence is the number of crops surveyed that have the disease. So if 2 fields out of 100 fields have a specific disease, then the prevalence is 2%, because the disease was seen in 2% of the fields surveyed, regardless of the incidence or severity of the disease. Incidence, however, is the percent of plants surveyed within each field that have symptoms of the disease. So if we say like 20% incidence means like overall 20% of the plants had the disease. And severity is the proportion of plant tissue that they showed disease. So to kind of understand how much disease we had, we should kind of consider all the three factors: prevalence, incidence, and severity, to realize like how much, for example, Sclerotinia stem rot we had on canola, not just looking at one of the three factors. So I will talk a little bit of what we saw for Sclerotinia stem rot on canola during the survey, during the general survey. Sclerotinia stem rot was present in 30% of crops that we surveyed, at least at trace levels. An average incidence of 12% in 12% of plants in infected fields had symptoms, with an average incidence of 4% in all fields. So if we consider all the fields and do the average, then this is the 4%. So it's across all surveyed fields, regardless of whether or not they had the disease. But if we just do the average for infected fields, then that's the 12% incidence in infected fields. And the same thing for severity. So severity was 0.1 average across all surveyed fields and 0.4 in infected fields. And again, the severity for the Sclerotinia stem rot is on a severity scale of 0 to 5, with 0 as healthy and 5 as plants dead. And then 2, 3, and 4 would be like 25% of the plant affected, 50% of the plant affected, or 75%. So when we look at all the 3 parameters, then we see that was for sure not a Sclerotinia year for Saskatchewan, as severity was very, very low. And it was, I guess, the lowest amount of Sclerotinia since past couple of years. So this graph compares average incidence of Sclerotinia stem rot seen this year and compares it back to previous years dating back to 1999 based on data from the General Canola Disease Survey. This graph shows how dependent Sclerotinia is on moisture and environmental factors. So if we look at this, we will see that, like, for example, Sclerotinia incidence peaked in 2016, which was a very wet year at almost 24%, then dropped significantly in 2017, which was a dry year, to 3.4% of incidence, and then it started to increase again and stayed at 17% in 2020, and then dramatically dropped in 2021 to a record low of 2% because 2021 was very dry, and then increased again in 2022 to 6% and slightly declined last year to 4%. So it's a very interesting pattern in terms of how dependent this disease with on moisture, and how its pattern is well aligned with the moisture we see in the province. And some of diseases are like this, so they love moisture, so the patterns are more aligned with moisture than some other diseases like blackleg that we shortly visit. So here's the regional breakdown of Sclerotinia prevalence and incidence results from this year's survey in the province. And prevalence was highest in the northeast and northwest regions, almost 53%, and the lowest in the southwest region, which actually we didn't find any Sclerotinia during the survey. Incidence was highest in northwest, almost 9%, and again lowest in the southwest regions, as we didn't find any. So blackleg is another very important disease that is assessed during the general cancer disease survey. Relatively has been less affected by environment relative to Sclerotinia stem rot. Even if it is a dry year, you still are able to frequently find it. This year, the numbers are very similar to last year. So 72% of the fields surveyed this year had at least terrace levels of blackleg. Overall incidence was 12% across all surveyed fields and 16% average for infected fields only. So very high level of prevalence, very high level of incidence, but severity was quite low with 0.2 on a scale of again 0 to 5. So it's quite a low severity when we average across all surveyed fields, and it's still 0.3 is also not very high, even if we averaged only for infected fields alone. And this is just the blackleg rating scale 0 to 5. So those numbers are quite on the lower side of the scale in terms of the severity. Regional breakdown for blackleg prevalence and incidence for this year. The Northwest had the highest amount of blackleg, with almost 90% of fields had at least a little bit of blackleg, 24% incidence, so 24% of plants had some level of blackleg. And Southwest had the lowest prevalence with 46% almost, and average incidence of 3% in the province. So Northwest highest amount of blackleg, and Southwest lowest amount of the blackleg. This graph is also an interesting graph, which is comparing blackleg incidence that was seen this year compared to previous years, going back to 2011. Overall, there had been an increase in blackleg being seen between 2011 and 2020. Incidence was quite high in 2020 at almost 15%. It's quite a peak there. And then 2021 was very dry, so it decreased, but not like as sharp as Sclerotinia stem rot. But again, increased in 2022 and 2023 to 11% and 12% respectively. So Aster yellows was a big surprise this year. We had like 10% overall, 10 times overall the amount of Aster yellows this year compared to last year. So if we only considering the 100 plants we assess in each field from the 5 surveying sites across that W-shaped pattern. So 33% of fields had aster yellows, but aster yellows is quite very obvious even from the roadside. So if we add those fields that they had aster yellows, but the aster yellows was not picked up in the 5 surveying sites within the field, so actually aster yellows was present in 65% of fields. So that 33%, which is the formal survey number for aster yellows being present in a field picked by the standard protocol of this survey, is very high. It's 10 times more than last year. And the incidence was 2% across all surveyed fields and 5% in diseased fields. So these numbers are very high for aster yellows. Usually, most people don't consider aster yellows among the big diseases like blackleg and sclerotinia stem rot or verticillium or clubroot. But this year it was not the case. It was important. And if we consider that when a plant has aster yellows, has almost zero yield, then like 5% incidence in diseased fields can translate to 5% yield loss. So powdery mildew is another interesting disease. We haven't seen it for a long time, perhaps for a decade or so. In 2021, there was a couple of calls from producers reporting a white clod at the time of harvest, and it was powdery mildew. And last year also a couple calls, but this year powdery mildew got picked up by survey, and it was seen in 13 of the surveyed fields with 6% prevalence, which is quite high. And so we are looking closely to what's happening for powdery mildew. It seems that this disease was not around and it kind of bounced back again. But the impact on yield should be limited for most cases. Moving to Alternaria black spot, like half of the fields were reported as to have Alternaria black spot by our surveyors this year. So this was were the results from the general canola disease surveys for more important findings, but applicable to canola, we have 2 separate programs as well, in addition to the general canola disease survey. So one is the Saskatchewan Collaborative Monitoring Program. So we go through it and quickly review what we found this year. So the Saskatchewan Collaborative Monitoring Program, of course, is a collaboration among the ministry with SARM Plant Health Technical Advisors, Saskatchewan Crop Insurance Corporation, SCIC, and SAS Canola, and to be successful in monitoring the spread and distribution of clubroot across the province and encouraging proactive on farm management. Last year, it was proposed with 5 components. And again, just a reminder that whatever we do in any pest surveillance in province is 100% permission based at this time, and then it also applies to the clubroot monitoring program. Clubroot monitoring program was also 100% permission based since last year. So last year, the clubroot monitoring program had 5 different components. So we looked at in-field surveillance in high clubroot risk areas, and we quickly defined that in the following slide. We also had in-field surveillance throughout the province, and we continued to try to increase external reports of clubroot infested fields by producers and agrologists. Still, that portion of the data is a big portion. So a main chunk of our data comes from external reports overall, encouraging the on-farm monitoring for the clubroot pathogen through the clubroot soil testing program, also known as SAS Canola program or SAS Canola clubroot DNA testing program or soil testing program, was also part of the was also under the umbrella of the clubroot monitoring program for the province. We also followed up on fields identified as positives during previous years' surveys. So collaborative risk-based survey in high-risk area is done through a reviewing of SCIC's data. So if a field is under intensive practice of canola cultivation, like at least 4 years in past 6 years, is included on that portion of collaborative risk-based survey in high-risk areas. But the change on this category from being conducted under the Pest Control Act to be moved to fully 100% permission-based has happened since last year. So this portion is also 100% permission-based. We also look at club root when we are doing the general canola disease survey, like any other disease survey, and it has been always permission-based. We do follow up on fields identified in previous years that we know that they have club root, but we also moved to a full permission-based survey even for this category since last year. Again, we also still continue to try to raise awareness and encourage external reporting. If the external reporting happens, we try to add some values to it. So we offer free pathotyping tests and free soil testing. Of course, free soil testing is also available through commercial labs, but pathotyping is only available through us because it is still a very labor and time and space demanding procedure being done in greenhouses only in some research facilities that we have the honor of having the support from University of Alberta over years. So we also have that farm testing program, also known as SAS Canola program. And we encourage producers to sign up either online or through SAS Canola or interested RM offices or through plant health technical advisors and test their soil. And it has always been permission-based as well. So this year, overall, we had 534 fields examined for club root in 205 RMs. We had 2017 fields examined for club root through the general canola disease survey. We had permission to collect samples for 182 of those fields and tested their soil as well. We looked at 188 fields in areas of the province where club roots or club root pathogens were known to occur through the club root specific risk-based survey, which is the one that we also look at the data, and if we see a field through the SCIC data that has 4 years of canola in the last 6 years, we include in that program. So this was great this year that we had almost 3 times of uptake compared to last year for volunteer soil testing program with the support from SAS Canola. And we followed up on 37 positive fields that they are known to us, and we had the permission to get back to those fields and check. So usually with the general locations of all these club root infested fields at the RM level, we used to generate the Saskatchewan club root distribution map to raise awareness and encourage proactive club root management and inform the biosecurity programs. And it's a very good guide to indicate which areas of the province have the highest club root risk. But it's important for producers to be proactive and scout their own fields to assess their individual risk because of the limitation that this monitoring program actually has in terms of the number of fields in total that we can examine. And again, since this is a soil-borne disease, the pathogen can likely be present in other areas or RMs near regions where club root has been confirmed. So in 2023, 2005 RMs inspected. We were fortunate to have seen actually to not actually see any fields with visible symptoms. So we didn't find any of those fields having visible symptoms, which was great for the province. It was not the case for other jurisdictions, so it was specific to the situation in Saskatchewan. There were just on the side. It's good to remind that many cases of club root visible galls were seen this year in other jurisdictions on the prairies. So DNA-based testing found one only one field new for Saskatchewan, which was translated to a new RM, which was RM of MacLeod 185. DNA-based testing or on-farm soil testing program found one field, but it was not new. And through the collaborative survey, we didn't find any new fields. 6 of the fields with positive history still they came back as positive, but they were not new. So the new findings was limited to only one from general Canada disease survey, which was through the DNA testing in the absence of visible symptoms, which was good news. We had 0 fields reported to us externally this year, perhaps because there was no development of galls, but overall still since 2017 till now, the big chunk of the data we have are from these external reports. So in total, we have still 82 fields confirmed in Saskatchewan with visible symptoms for clubroot since 2017. So there was no increase from last year to this year. With one field with DNA of the pathogen this year, the total number of fields with DNA but with no visible symptoms increased from 42 to 43, so only one field added. Again, this very small table is the history of clubroot in Saskatchewan, how the number added up, and we got to 82 fields with visible symptoms and 43 without visible symptoms, but only with the DNA of the pathogen. So overall, 30 RM in Saskatchewan have the history of visible symptoms, and 21 RM they have the history of DNA but no history of visible symptoms. So 51 RM in total, they have some kind of clubroot presence in their areas out of 296, which is actually a success story for clubroot management. We know the impact of droughts, but drought was for everywhere across the prairies. So also I interpreted somehow as a success story in terms of its management. But we should continue to be very cautious on this sort of soil-borne diseases. Pathotyping side of this stuff, of course, we had no visible galls this year, and there was no pathotyping for sure because there was no visible galls to test for. But so far from 2022, 2021, 2020, we have no finding of any of those pathotypes that they are able to overcome resistance from the pathotyping assays done at the University of Alberta. So another separate program we have for monitoring of a specific disease is the verticillium stripe monitoring program. At this time, I guess Saskatchewan is the only province that has such a program to specifically look for verticillium stripe as a separate program on top of the general cannula disease survey. So I'm going to, because everything is quite known or clear for other diseases, they are well advanced in research. But when we move to verticillium stripe, there is still lots of unknowns and world of ambiguity and still a lot of good research in progress. But overall, it is a disease that first found in Manitoba in 2014, and then CFIA looked at the across Canada to find the pathogen through DNA testing. They found it in Saskatchewan as well as other jurisdictions. So we knew that we had the DNA, but we have never confirmed the disease with visible signs in commercial fields since 2021. It seems that it poses a new threat, threat to the brassica crops in Saskatchewan. Excuse me, and across the prairies. It seems to be a main disease. Sometimes we mention that it is another big disease, so it's not a minor disease. It is perhaps it changes the 3 big diseases of canola, blackleg sclerotinia, and clubroot to 4 diseases in terms of its impact. Again, like clubroot, it's a soil-borne pathogen. still much to learn. Lots of good research is being done now, and since it is quite new for the prairies or in Canada, still most of the data that is being discussed come from European literature. We wait for our researchers to provide us with solid results, and because most of the research haven't been started till 2019, we are looking forward to perhaps late 2024 or early 2025 to listen more, to hear more from our researchers across prairies on different aspects of this disease. Usually, just for this specific disease, I added some symptomatology slides because it's still new. So if you want to identify it in fields, there are multiple symptoms, but just for the interest of time, if we see stripes up one side of the stem, and if we see if like this picture, stripes up one side of the stem, this is the reason we call it verticillium stripe. And also if toward the end of the season, close to harvest time, if the epidermis or the skin of the stem peels back and reveals very many tiny structures called macrosclerotia. These are two of the three big symptoms of the verticillium stripe. So stripe up one side of the stem toward the harvest time or shortly after harvest time, the epidermis gets peeled back and shows lots of very tiny structures called macrosclerotia. Again, more pictures of epidermis being peeled back and showing those tiny structures, pepper-like structures called macrosclerotia. And the third important symptom is having this gray hueish pattern. If you get your clippers and cut the stem at the soil line a little bit through the root system, you will see a gray hueish, sometimes starburst pattern, which are actually the fungus for verticillium stripe. And if you compare it to blackleg, you will see for blackleg we will have more solid black wedges, while for verticillium stripe we have this hueish pattern. And of course, the size of macrosclerotia on the stem for verticillium stripe is much smaller than pycnidia of blackleg. And again, this is a great picture from Canola Council from Justin Carnelson to show both diseases on the same plant and show how is the size difference between the pycnidia of blackleg versus the macrosclerotia of verticillium stripe. If you want to just know what's going on in the lab, when we send the stems, they're going to be plated. The black fungus is the fungus that is the verticillium longisporium. Then the lab does the microscopy and tries to differentiate different species of verticillium that they can be involved in the disease. So it can be verticillium longisporium, which is the main pathogen, but it can also be other species like verticillium dahlia. And there are two different set of PCR. So one is for genus that we make sure it's verticillium, and then for a species that if it is verticillium dahlia or verticillium longisporium. So our journey through this disease actually started from 2021, so very recent. In 2021, 4 suspicious samples were found and were sent to the University of Manitoba. 3 came back as verticillium dahlia, 1 as verticillium longisporium, lineage A1D3, which was new for Canada. Then toward the end of the season, a farmer who is also a plant health technical advisor sent 2 more samples, and they came back from the lab again as positive for verticillium longisporium. 1 was A1D1, 1 was A1D2. So we know that we have all the 3 lineages of the verticillium stripe in Saskatchewan, and it questions actually that if the disease was actually really new or is actually really new, how then we can have this amount of diversity in just 6 samples. Again, lots of unknowns, lots of studies going on. So 2021, we confirmed we have the disease with visible signs and symptoms, and the picture you see is actually the sample, the real sample that was found first in Saskatchewan. We know that we have high diversity of this pathogen in Saskatchewan. So in 2022, we found only 1 sample of confirmed for verticillium stripe through the general cannula disease survey. So people was asking if the disease is still rare. So in Saskatchewan, so we put together a very small scale proof of concept type of survey, and a small group of us looked at the areas close to Manitoba border just for this disease. Again, we found the field that was good in terms of learning for and training eyes to see verticillium stripe on the east side of the province. We got trained there with the whole group. We moved to the far east of the province, looked at 25 fields in 2022, and we found like 15 of those 25 had some sort of suspect samples. So they had, like, for example, a stripe and stuff. So they had suspect symptoms, but then they went through the DNA analysis, and like half of those fields got back as confirmed. So 28%, like 7 of the 25 was confirmed to have verticillium at the genus level, and only 3 was confirmed to have verticillium longisporium, which can translate to 12% of field with verticillium longisporium. So we are learning. We are also in our learning curve, and actually we, I guess I assume, to the best of my knowledge, we were the only province who did this, like did genus confirmation and then genus species. So we provided this information to other interested groups, so they also know that other verticillium rather than longisporium can be involved in this disease. So 2023, we had the experience from 2021 and 2022, so we decided to expand. So in addition to general Canada disease survey, we decided to expand the verticillium stripe after harvest specific survey to the whole province. We look at more than 100 fields this year, and we tried to choose those as a subset of fields that we already surveyed for general Canada disease survey. So general rule was we do 200 for general Canada disease survey, and then we do half of those for verticillium stripe specific survey, because we want to make sure that we have a good understanding of the risk to the whole cannula community in Saskatchewan, not only on the east side. Of course, the infield portion of all the surveys completed, but lab analysis for verticillium stripe specific survey is still going on. It's a time-demanding process in lab. But we have results from general Canada disease survey for verticillium longisporium. It was confirmed in 23 fields only through the general Canada disease survey. So quite high number for us, because it's 11% of the fields from the general Canada disease survey. And again, this is a map that Carter actually prepared with this manually marked in blue, those RMs that we found verticillium stripe through the general Canada disease survey. We also had one finding out of the survey in Indian Head area for the general Canada disease survey that we're going to add to this map. But the more comprehensive map will be generated after we receive the results from 102 fields with that we surveyed after harvest through the verticillium specific survey. The DNA analysis is still in progress at the U of Manitoba for this portion, and we'll provide it to the community as soon as we have them. So moving to again, cannula portion was took me longer to cover, because in addition to general cannula disease survey, we have 2 separate programs, which is collaborative monitoring program and the verticillium specific monitoring program. But we also do general surveys for other crops, and they are very important to us. So we have 3 different surveys that we do at the ministry with our counterparts for lentil, field pea, and soybean. And we looked at 54 lentil crops, 42 field pea, and 20 soybean this year. Moving to lentil diseases, again, 54 lentil fields were surveyed between July 10th and August 9th. 100 plants per field, close to the pattern we use for general Canada disease survey, along a W-shaped pattern. And we'll look for diseases such as anthracnose, ascochyta, sclerotinia, botrytis, staphylium, and different root rot complex in lentil fields. Again, the same thing applies here as well. The prevalence is the number of fields with disease, incidence number of plants within each field with disease, and severity, the proportion of the plants diseased. So anthracnose remained the most important foliar disease in lentil. It was present in 74% of lentil crop we surveyed. Excuse me. Sorry, I have like a bad throat. I guess because of talking too much. And excuse me. So anthracnose was again present in 74% of lentil crops in the province. Average incidence was 20%, and severity was quite low compared to previous years, with 0.3 out of 5 overall and 1.3 out of 5 for infected plants. Staphylococcus blight is an interesting disease. We closely monitor this disease, and research started to investigate the host range for Staphylococcus blight pathogens involved in lentil, with other Staphylococcus issues that we started to observe in province. So Staphylococcus blight was observed in 20% of crops this year, with an average incidence of 4%. Across the regions, the prevalence of Staphylococcus blight ranged from 0, like East Central and West Central. We didn't find Staphylococcus, but in Southwest, we had like 8% of our crops with the disease. And all of these numbers are lower than last year. So last year was quite half of the fields had the disease, and average incidence was quite similar, but way more fields had the disease. Sclerotinia stem and pod rot was observed in 6% of crops, average incidence 0.1%, so definitely not a big issue for most fields. And in fact, we found it only in 1 and 2 fields, each of the Southwest and West Central regions. So 1 field in Southwest, 2 in West Central regions, not a big disease for lentil this year. Ascochyta is another interesting story, I can say. It is quite a couple of years that we found Ascochyta blight is now hard to find, so it was quite rare. This year we found it only in 4 fields in West Central and like in Southwest, and 4 fields in Southwest, 1 field in West Central. Average incidence was 2% in fields with affected plants. And on the other hand, it is important to note at this stage that diagnosis is only based on visual symptoms in the field. So plants that having visual symptoms that they're suspect and were consistent with Ascochyta blight were not further confirmed with additional testing in the lab. So again, it is 2 years that I guess it is perhaps because of the good resistance we have that finding a typical symptom of Ascochyta on lentil is quite hard. So root rot is still a big issue. So root rot complex symptoms were present in 74% of surveyed crops across the province. The severity was 1.6%. And across the regions, prevalence ranged from 61% to 100%. And average incidence was again 35% overall. And high level of root rot was seen in West Central and Northwest this year. So all of this data that I am presenting today has been posted on Western Forum on Pest Management website. So if you go to Western Forum on Pest Management website and go to the reports for 2023, you will see all these reports are posted there. So if you have interest in knowing more on a specific region in the province and what was the level of different diseases for that specific region of interest, then you can go to Western Forum on Pest Management website and download the report and look at the data. But for the lentil, for sure, root rot and anthracnose was big problems, while Ascochyta, Sclerotinia, and Botrytis, they were not big issues this year. We had zero finding of Botrytis. Staphylium, again, because of its patterns on multiple crops, is of interest and is under close monitoring. So again, incidents, all these data as mentioned are posted on Western Forum on Pest Management. Again, in addition to prevalence, you can also look at incidence for any specific region. Again, incidence was this year quite high, again for root rot and anthracnose, but perhaps lower for other diseases. I move to field pea diseases now. We also surveyed 42 field pea crops this year, and between the June 28th and July 27th, and we look at 50 plants in each field for this specific survey. Try to record what we find. We can find unimportant diseases like root rot, Mycosphaerella, Ascochyta blight complex, dunny mildew, white mold, bacterial blight, and we also do the severity assessment for root rot and Mycosphaerella Ascochyta blight complex. Again, all the assessments are based on the visual symptoms in field. Mycosphaerella Ascochyta blight complex was present in 90% of the surveyed fields, and average incidence was 55% this year, ranged from 35% in northeast to 75% in northwest, and average severity was quite low at 1.8 across the province. So very high level of prevalence and incidence, but quite low level of severity, perhaps because of the good resistance and good management. And again, this specific severity scale has it starts from 1, with 1 as a healthy plant, so 1.8 is quite low actually. So interesting pattern in bacterial blight, and in 2024 we will closely monitor in general what's going on specifically for bacterial blight issue in the province, but we will see symptoms consistent with bacterial blight more frequently for field pea in general, all across multiple crops. Specific to field pea, and during the general field pea survey, we found these symptoms in 40% of crops, and average incidence was 8%. And bacterial streaming test was conducted with a microscope on representative samples from a portion of assessed fields. So it was confirmed on those, so we trust on our data for bacterial blight. Presence of this disease may be influenced by crop damage due to adverse weather in this region. So in fields with more environmental injury, we had more bacterial blight. Dunny mildew was a big issue last year, like what last year was present in 40% of fields. Average incidence was almost 11%. This year was drier, so that was not a big issue, I can say, in most cases. But it was present in low level in 17% of fields. Average incidence was 1.4%. Root rot complex, again, a big issue in general for pulse crops, was present in 90% of fields. Average incidence was 62%. Severity was 2.5 out of 7 overall, and 2.7 out of 7 for infected fields, if we average only those fields, for infected plants in infected fields. So average disease incidence ranged from 23% in northwest to 82% in southwest, but again, disease severity was generally low in most fields this year. But again, overall, a very, very big issue. So again, we also have the regional breakdown of all the data for prevalence and for the incidence for field pea as well. Again, root rot and Mycosphaerella Ascochyta complex were the two main diseases, while we find a lot of bacterial blight. So similar to prevalence, we also have tables for regional breakdown for incidence. Again, root rot complex actually had the incidence of 62%, and microspheral had the average incidence of 55%. So two big issues for sure for field P, in addition to bacterial blight that had 8% incidence overall across the surveyed fields. Soybean, we also have a very small survey for soybean. So we look at 20 soybean fields. We looked at them from August 2nd to September 6th this year. 19 of those were in Southeast and one was in East Central, and we look at 50 plants in each field. We look for all the important diseases of soybean. We also send the roots of soybeans to Dr. Yang Min Kim to check for root rot pathogens, but particular interest is on Phytophthora. So just a very quick summary of findings for soybean, and those numbers in bracket are from last year just for the comparison purposes. And again, the most prevalent disease in Saskatchewan soybean was this year was brown spot, but bacterial blight was also seen in almost all fields surveyed, and 79% incidence was for bacterial blight, 86% for brown spot. So this pattern hasn't changed a lot over years, and these two diseases remained the most important diseases of soybean in the province. So brown spot and bacterial blight. We had reports of symptoms that they looked like Cercospora leaf blight, which is different from frog eye leaf spot, which is also caused by a different species of Cercospora. But none of those were confirmed at the lab. So still, we have the bacterial blight and brown spot as the two most important diseases of soybean in the province. Just to touch on our program for Fusarium head blight, I have only 3 slides, but that's a huge program itself. Still, the analysis of the samples being continued at the crop protection laboratory with new testings in place in terms of the DNA analysis of those samples, which is great to have those testings in the lab, thanks to the crop protection laboratory team to add this testing, and they're optimizing would be very, very helpful. But just touch base on overall results for Fusarium head blight. This year we had 118 quality samples. We had more, but some of them, they were too immature or they had issues. So we had 118 quality samples to check for Fusarium head blight. 70% of barley samples, 86% of common wheat samples, and 52% of durum wheat samples had at least one suspected Fusarium damaged kernel. For barley, FHB was found to be the most prevalent in the gray soil zone this year, with at least one FHB infected kernel observed in 82% of the fields surveyed. Common wheat had the highest FHB prevalence in the black soil zone at 96%, while durum wheat had the highest prevalence in the dark brown soil zone with 58%. So the overall average preliminary severity rating of fields with observed Fusarium damaged kernels were 2.3% for barley, 0.2% for common wheat, and less than 0.1% for durum. And the highest FHB severity rating occurred in the black soil zone for barley at around 4%, the gray soil zone for common wheat at around 3%, and both soil zones sampled for durum wheat had very low severity. And I assume this was my last slide. So, so if you have any questions, you can contact myself or Carter Prue, our integrated pest management specialist that coordinates most of these areas, excluding the collaborative monitoring program, or Raul Avila, our pest regulatory provincial specialist, who has the collaborative monitoring program under his portfolio. Also, you can ask any questions at any time from our great crop extension specialist team, and you can, as Queen explained, you can find your crop extension specialist in your area and send your questions that way. So I am done, and thank you very much for listening. And I'm happy to have the opportunity to share these results. And the stage is open for any questions.
Thanks, Alireza. Yeah, we have a question here. So what is the easiest way to tell the difference between Microsphaerella blight and bacterial blight?
Yeah, so on field P, we have these two diseases, Microsphaerella and bacterial blight. There are differences that we can quite carefully differentiate the two diseases. Like bacterial blight, for example, has these. If you look at the leaf, if you are in the field and we look at the leaf through the sunlight, and if it is translucent, then it's more likely bacterial blight. But if it is like brown, dark brown, or quite dark, close to black, or even dark purple flecks, then it should be Microsphaerella. And if it is absolutely round and it has pycnidia on that, on a circular pattern or concentric pattern, then it is Ascochyta. But during this survey, we don't want our surveyors differentiate Microsphaerella versus Ascochyta. This is the reason we call it Microsphaerella / Ascochyta blight complex. But to differentiate that complex from bacterial blight, then there are some characteristics that mentioned on different websites to differentiate the two. But the translucent the translucent look of the bacterial blight through looking at the sun at the field would be very helpful.
Thank you. Yeah, as a reminder, if you have any questions, please put them in the chat box. We got another one here, Ali. So with verticillium being a newer disease, what are the best symptoms to look for and best timing? How can we confirm it's verticillium stripe?
Yeah, that's a very good question. And the problem with verticillium stripe is it comes early, but we can't see it. Macrosclerotia germinating soil at the flowering time or before that, and the pathogen gets to the xylem, but we don't see anything. When we see it, it is too late. So it is not like a disease like powdery mildew that comes late, so we see it late. This disease is very different. It comes early. We don't see it until it comes late, and it is around the harvest time or shortly after. So the best time to check for verticillium is closer to the harvest time, toward the end of the season or shortly after harvest, and look for. There are multiple symptoms that we can look for and we can name, but just to the interest of the time, I focus on the three most important. So one is seeing a stripe up one side of the stem. The second one is stem epidermis or stem skin being peeled back and shows us microsclerotia, which are like those very small pepper-like structures. If you have you need to have a good magnifier to be able to see that. So they are that tiny. They are much smaller than blackleg pycnidia. So you need a good magnifier to see those macrosclerotia. Also, the third symptom is when you get your clipper out and cut the stem at the soil line a little bit through, or perhaps take the plant out and cut the plant at the soil line a little bit toward the stems, a little bit toward the root system, and look at those cross-section pattern to see if it is the hueish starburst pattern versus the solid black wedges of blackleg. And then, of course, you don't look at one symptom, you look at all. So if you see the stripe is there, the epidermis is being peeled back and macrosclerotia you see, and also you see the hueish pattern or starburst pattern at the cross stem, then it's verticillium stripe. But the issue with a stripe and the hueish pattern in the cross section is other diseases also share those two characteristics. The only feature of this disease that is specific only to verticillium stripe is macrosclerotia. So if you see the epidermis gets peeled back and you have a good magnifier and you will see macrosclerotia, then that's the best, because this disease doesn't share this characteristic with any other diseases. While stripes and hueish pattern in cross section are good symptoms, but there are other diseases like fusarium wilt that can share those two specific characters or characteristics.
Okay, there's another question in here that says, with it likely being another dry year in 2024 growing season, what diseases do you still need to keep an eye on in dry conditions?
Yeah, so that's a great question. So almost all of these diseases are very well established in the province. So we may see all of them, but only the level may go down and up a little bit, depends on what pathogen actually is, and if the pathogen is relying on moisture. So if we get a drier year, then we'll see for sure like less Sclerotinia stem rot, because that pathogen is totally relied on moisture. But there are pathogens like and diseases like blackleg that they are less affected by environment. So my outlook for 2024 is we will see the same amount of blackleg as 2023 and 2022, because the pathogen is not going to be affected by the level of moisture so badly or so significantly. The situation for Verticillium stripe is very different, because the pathogen likes hot and warm, hot and dry conditions. So if we get another hot and dry condition, then we may even see more Verticillium stripe, because that's a different pathosystem and it thrives more in hot and dry weather. As soon as the plant is under stress, it is easier for Verticillium to get to the xylem or to the vessel of the plant and occupy that and multiplicate that and clog the vessel or xylem. So as the plant is more under stress, Verticillium is happier. So it depends on the disease. It may be different, but this disease is unfortunately, these diseases we talked about about today are well established. So we'll see them, but depends on how much moisture they like. We'll see a little bit more or less of them. But I really hope we don't see Cloverwood again, because I was very, very happy to not to see Cloverwood symptoms this year in Saskatchewan. So I hope that we don't see it again in 2024, but no guarantee, because it's being frequently found in other jurisdictions like Alberta, close to us.
Well, thanks for that, Alireza. That appears to be the last question. Just a reminder, if anybody has any questions, to reach out to your crop extension specialist or Alireza. We'd be happy to answer any questions that you guys have. We're gonna go back to James and test his audio here first, just to make sure that everything's working.
Right.
And if it's not, then we will switch to another date.
Can you hear me all right?
Yeah, we can hear you, James.
Okay. Let's see if I can try to if I could get the link to share my presentation.
I think that Ali's just gonna send it to me, and then we'll just do what we were doing before. Okay, we should be good to go.
Okay, excellent. Yeah, let's get right into the first slide, please. So first thing we're going to talk about is grasshoppers, and apologize for the garbled sound on my previous go with this. So looking at things from 2022 and 2023, so 2023, of course, this past year, you can see that there is a relatively marked increase in grasshopper populations year to year. Of course, we're experiencing a relatively protracted drought period, and that really favors grasshopper population increase. If we could go to the next slide, please. The primary pests that we're going to talk about, there are 4 primary pests. The most prevalent in Western Canada currently is two-stripe grasshopper, which is a bit interesting considering all documented previous outbreaks or major outbreaks of grasshoppers associated with drought conditions have been of migratory grasshopper. Oh, we lost the video momentarily. Can you see it again?
Yeah, I think we're there.
Okay, excellent. So yeah, once again, the previous outbreaks have been associated with migratory grasshopper, which is another species. Not to say that other species don't engage in migratory behavior. This one certainly does. You can see what's characteristic about these ones, especially the adults, the two stripes extending from between the eyes to the tip of the wings there, as the wings are folded over. Thus, two-striped grasshopper. These are large animals, so 1.5 inches plus for females. Females are larger than males, which is the usual case in grasshoppers. Males don't fight amongst each other for territory, which is usually the selective pressure that leads to larger size in males. So yeah, females arguably greater contribution to the following generation, and can be very fecund. So that is, can produce a large number of eggs, 200 plus per female. Certainly not uncommon for a number of these species. So you can see the potential for rapid population increase. In the case of two-stripe, and in the case of all of the pest grasshoppers that we deal with in the province, they all overwinter as eggs. So they need a cold period to stimulate their diapause, and then a warm period in the spring to come out of diapause. Diapause is a state of arrested development. We'll talk a little bit more about that in an upcoming slide. Pardon me. These two-stripe tends to come up ahead of migratory grasshopper by almost 2 weeks, and in the case of 2023, because of the warm temperatures, we had occurrence of these animals at least 2 weeks early in a number of different parts of the province. So that is late April, these animals were coming up as nymphs and causing economic problems or requiring insecticide application in some parts of the province. The really conspicuous state is that fourth instar nymph. If you can get it at that state or before that state, you have a good chance of really doing a good job on that population, and it really opens up your opportunities for different control products. So bright green and that characteristic black stripe on the hind femur, you see that in the adult, you see that in all stages of this species. As a rule, this one prefers lush habitat and lush foliage, but it will feed on cereals if push comes to shove. Not its preferred host plant, and it actually pays a bit of a price for feeding on cereals exclusively. When you're looking, pardon me, I did mention these do engage in migratory behavior, and you can see that the range of these animals extends over the bulk of North America. So particularly in regions that are characterized by dry conditions and with limited resources or concentrated resources, you'll see this migratory behavior. They will engage in it in this part of the world, and I certainly witnessed that, and I'm sure many of you witnessed that driving on some of the highways in the province. Next slide, please. So migratory grasshopper, this is the one that is typically the dominant one in grasshopper outbreaks, or has been historically. So a bit of an oddity why we're seeing two-striped grasshopper as the dominant right now, and in short, I don't know why. But migratory grasshopper is still dominant in some regions of the prairies, although it's they tend to be more restricted to the south. Characteristic features of these ones, looking at the adults, you can see the blue arrows. So those splotches on the hind femur, very characteristic. The red hind tibia can be characteristic, but there are other species that have that. And that little camo stripe on the hind wing. So you get those three characteristics, plus a relatively smaller size. The females in this case are about 1 inch, 1.2 inches long. Two-striped grasshopper, of course, 1.5 inches or larger. They make up for their relatively smaller size with voracious appetites and large numbers in some cases. Once again, highly fecund, 250 eggs per female. They do start to hatch in areas with greater heat accumulation. So everything about their development and their physiology and their behavior is dictated by external temperature. They're ectotherms. Unlike us, we're endotherms. We control our internal temperature physiologically. They're reliant on the external environment and how they can modify that behaviorally. So where heat is accumulating more rapidly, we're going to see rapid emergence of these nymphs in the spring. Relatively characteristic, you see that dark stripe extending from between the eye. relatively characteristic of migratory grasshopper nymphs. They do have a rapid development time and can go from emergence of nymphs to adulthood in about a month, under very warm conditions, and we certainly saw that this past year. Really rapid hatch, highly numerous hatch, and rapid development through nymphal stages to adulthood. Why that's important is the faster they can get those eggs in the ground, the more heat that those developing embryos in those eggs can accumulate. They need to get to a certain point of development to overwinter successfully. That is, there's a cold hardening period or a cold hardening state, and once they get to that state, they become very cold resistant. And they need temperature to do that. So broad host range in the case of these guys, real garbage guts. Really, if it's green and it's not nailed down, they're going to at least give it a go. So this includes, even coniferous trees and cactus, other insects, both alive and dead, dung. Once again, if it's organic and push comes to shove, they will take advantage of a broad assortment of different food sources. Next slide, please. Sorry, I'm hitting page down and nothing's happening. So the third species we'll talk about, and this one is usually number 2 in outbreaks, but it's become a distant third this time around. Another large animal. These ones are again 1.5 inches plus as females. Similar in appearance to two-striped grasshopper, but you can see the 2 stripes on the pronotum, so that plate right behind the head, don't extend to the tip of the wings. So why the differentiation is important is while two-stripe has a relatively broad host range, Packard's also has a relatively broad host range, but it's more narrow, and they really do prefer legumes. They also have a real predilection for thistles, and will happily munch on thistles, if available. Their nymphs are very characteristic as well. That bright green, they can extend into yellow, but you can see that there's no stripe on the hind leg in the case of the nymphs. So the adults also have blind or blue hind tibia this is subject to some variability though. But the characteristics of that 2 stripes on the pronotum coupled with a blue hind tibia and that large size, and in the case of the nymphs, the bright green coloration gives them away. So these ones do prefer open habitat and light textured soils. And greater pressure typically in northern regions, although you can see the range extends over the bulk of North America and throughout the Great Plains. And as I mentioned before, their host preferences as well. Next slide, please. This is the 4th important pest species. It has been numerous in some locations on the prairies, although the numbers are down considerably, even in the last couple of years. For this one, this is clear wing grasshopper, very characteristic camouflage pattern on the hind wing. You can see there are carinae or little keels. You really got to get pretty close to see those ones, and it's a relatively small grasshopper as well. So about 1 inch long, 1.1 inches long for females. But that we'll say medium size coupled with that camouflage pattern really gives them away. And you can see the nymphs, or at least the first instar nymph, very characteristic with that black and white pattern. So if you see them coming up, then you know that you're dealing with clear-wing grasshopper. They also engage in migratory behavior, and in some parts of their range, primarily the West Coast, they're called California migratory locust, or inland California at any rate. They do like to emerge with conditions that are conducive to the growth of green plants, or the rapid growth of plants. So in the morning, especially after an evening shower, they are primarily a grass feeder, and you can see these animals in large numbers in broadleaf crops, not causing any damage. What they will do on occasion is sun themselves on surfaces, so they're functionally pointing themselves broadside at the sun to increase their internal temperature. They like to get their internal temperature into the high 30s and sometimes in the low 40s, depending on what sort of needs that they have. But yeah, these the pest grasshoppers really like it warm. Next slide, please. So an important consideration is that most of the grasshopper species on the prairies are not pests. So if you've got long antennae, these aren't grasshoppers at all. And you can see that the green fella at the bottom there, that's actually a meadow katydid, which is they cause the male chirping is that long trill that you hear in hayfields. These are not typically damaging economically, although we did have a couple of spots in Manitoba where these were reported as damaging in hay. So they can be very numerous without causing economic damage. So typically not something that you need to worry about if you see them in a hayland situation. The one directly to the left of it, that's what's called a speckle-winged rangeland grasshopper. And over to the other side of the meadow katydid, you can see the animals with the red wings and the yellow wings. These are different color morphs of this speckled-winged rangeland grasshopper. These overwinter as relatively mature nymphs, so they'll achieve adulthood relatively early in the year and become very conspicuous. They're a decent sized animal, you know, about the size of a migratory grasshopper, so about 1.2 inches, and very noisy flyers. And so they're very conspicuous. They got the bright coloration, but if it's got wings in the spring, it's not a pest. If it's, or wings before late June, not a pest. Brightly colored hind wings, not a pest. And that includes the Carolina grasshopper, which can be relatively spectacular. I mean, it's a big animal, noisy flyer, a bright yellow band on the hind wings. Typically not a pest. The vast majority of grasshoppers on the prairies are herbivores and are happily munching on native plants. So they're really not much of an issue in crops at all. So callers, chirpers, noisy flyers, wings in the spring, not pests. Next slide, please. So populations and weather. I'm sure this is the topic of much discussion right now. And in the case of migratory grasshopper, and this is what the models are based on its prevalence as the dominant species in previous outbreaks. So what's said for them is true for the other ones, you know, in broad strokes. So warm spring, earlier hatch, and we certainly saw that for two-stripe, increased nymphal survival, increased fecundity, so increased eggs per female, more rapid development of all stages. And that warm late summer and fall, that's really important for oviposition or getting the eggs in the ground. And as I mentioned before, the embryos need to get to a certain point to overwinter, to harden off for the winter. And there was plenty of time and really ample conditions to do that in this late summer and fall. And warm, dry conditions promote embryonic development as well. Next slide, please. So I'm not sure that everyone has an appreciation for what grasshopper oviposition looks like, or really what I'm getting at is how well protected these eggs are. So moving from the top left, you can see the happy couple. And that, of course, leads to fertilized eggs, which are deposited into the ground. So the female is pushing her abdomen into the ground using both muscular strength and hydrostatic pressure. And so she can get that, she can extend her abdomen a fair ways, almost 2 inches in the case of large grasshoppers. She's going to lay those eggs and secrete a foam. And you see that lower left, and that foam hardens like cement. So it's very, you know, the depth that the eggs are laid in the soil, so the soil and the moisture in the soil lends to temperature stability and protection from temperature extremes. You get some snow on top of that gives additional protection, and this hardened case around the eggs also gives protection from environmental conditions and predators. And finally, just a photo of a two-striped grasshopper with her abdomen sticking into a crack in the ground there. Next slide, please. So looking at conditions in the fall of this past year, so this was from August 31, so late summer, fall, relatively widespread, very dry conditions in that time. What this means is the conditions were conducive for egg laying. Large numbers of grasshoppers laying large numbers of eggs is what we're looking at. So there is a pretty significant bank of eggs in the soil right now. Can we go to the next slide, please? So if we're looking, this is the grasshopper oviposition index. This is developed by Canada. And these are conditions under normal conditions, or this is what oviposition would look like under normal conditions. And you can see in that Palliser triangle area, of course, oviposition is typically relatively high. Extending throughout the south. If we can go to the next slide, please. This is what things look like this past year. So there are a lot of eggs in the ground, a lot of eggs in the ground. We've had some questions about cold, the sudden cold, hard cold snap without snow contributing to grasshopper egg mortality. And yes, it is a possibility. They are still pretty cold hardy, and I think a consideration is going to be for the numbers of eggs that are actually in the soil. So even if mortality is relatively significant, we're still looking at a lot of eggs. And so we're very likely looking at significant grasshopper populations coming up in the spring. Next slide, please. So temperature, very important, fundamental. So diapause, as I mentioned before, required before some can complete development. They can get through their development relatively quickly and be in a diapause ready state in the case of migratory grasshopper at 30 degrees C in about 2 weeks. So those embryos need to get to about 80% development. So this means they're almost ready to go when the spring, when the warm up in the spring occurs. They only got 20% to go. They can get through that in a matter of days. So they can be relatively early emergers. Two-stripe emerges even earlier. In the case of clearwing grasshopper, which is one of the later ones to emerge, they're ready to go in about a week, but they're only 50% developed. So it takes that development a little bit longer, typically a couple weeks longer in the spring to come out. So they need those warm conditions to break diapause. And when we're talking about diapause, it's not hibernation. So hibernation is typically what's considered cold temperature torpor, or this slowing of metabolic functions. In the case of diapause, it's a full arrestment of development. So development stops, and really that's what diapause means. So they'll enter this quiescent stage and wait for the warm temperatures to stimulate them to complete development, and then they hatch. And sometimes they can be very numerous when they do so. Next slide, please. So I mentioned before the developmental stage, and it's important to note that it can actually be too dry for grasshoppers. So the eggs must actually absorb water, if there is inadequate moisture for them to complete development, especially in the early stages, they can arrest their development and stay in a state of delayed diapause for years if they have to, waiting for appropriate conditions. So we can get this synchrony of emergence of not necessarily last year's grasshoppers, but also the previous years and even years previous to that. If conditions are conducive for continued development to hatch. So 13.5 is the magic number for migratory grasshopper. For two-stripe grasshopper, embryos will actually die if they lose 2/3 of their moisture. The egg case, or the chorion, however, is relatively watertight, and it takes quite a bit of dry conditions, very dry conditions, for them to lose that much moisture. And in the case of clearwing, and I think the same could be said for all of them, soil moisture, of course, is going to have a buffering effect on soil temperature fluctuations, and that's going to help with cold hardiness as well. Next slide, please. Oh, sorry, actually, if we could just go back to that one, just for a point of comparison, if you look at stage 24, so that's where migratory grasshoppers at. So you can see those embryos are really well developed. And in the case of clearwing, that's about stage 19. And so yeah, they're recognizable as an insect embryo, but only just. They haven't even reached blastokinesis at that point, where they do their little acrobatics in the egg. Next slide, please. So cold hardiness, there can be significant egg mortality at -15 degrees Celsius for migratory grasshopper. A thing to consider for that is that these are eggs exposed at ambient temperatures. So these are not eggs in the egg case in the soil covered with snow. So they can suffer significant egg mortalities at -15, but you can see the difference 20 centimeters of snow makes. And of course, the deeper the snow, the greater the insulative effect, and you can get close to 0 degrees at the snow with good snow cover. The more advanced development, as I mentioned before, 50% for clearwing and 80% for migratory, they become quite cold hardy. Next slide, please. When we're looking at clearwing, you can see, and unfortunately the line doesn't show up very well, there is a squiggly line that is relatively flat, going from 40 down to about 30% at -23°C. And that's at ambient temperatures for a month. So they are pretty cold hardy. -29 seems to be a pretty good cutoff for them, which leads to mass mortality, but of course that doesn't factor in the protective effects. Next slide, please. High temperatures can kill nymphs. So very warm soil surface temperatures can cause difficulty in molting for clearwing. So if you think of, most people are familiar with what insect molting looks like, it's really the exchange of one suit of armor for another. They can get locked in the previous suit of armor if conditions are too dry, and die under those conditions. In the case of migratory grasshopper, of course, soils can really warm up. So at 35°C, that soil is going to be into the 40s, and that can lead to nymph mortality. Adults are tougher though. I mean, they can take ambient temperatures, high 30s, low 40s, and take those for some time and be very, very active. Next slide, please. So grasshopper outbreaks in the 30s, late 40s, early 60s were all terminated by cool, wet weather and the outbreak of a fungal infection called Entomophthora grillii, or summiting disease. And this is a picture I took down near Weyburn. This is a clearwing grasshopper, and you recognize that, of course, by the camouflage pattern on the hind wing. That was obliged by the fungal pathogen to climb to the top of a plant, and the suit of armor analogy applies here again. So that suit of armor locks in place, and animal dies, and fungal spores are shed from the rotting tissue inside the suit of armor. So those fungal spores are going to rain down in the environment, and they're going to land on brothers and sisters and pals, even loose acquaintances. But yeah, it's a good way for the fungal infection to spread and become epizootic. So next slide, please. So looking into the crystal ball for the upcoming season, temperature and moisture, highly influential, as I mentioned. So this is from Environment and Climate Change Canada, or ECCC as it's colloquially known. And this is the seasonal forecast into April of 2024. We're looking at a high probability of increased temperatures. It doesn't look like we're going to suffer as much as say Labrador or the West Coast with very warm temperatures, but definitely a higher probability of increased temperatures. That's going to lead to increased development, greater embryonic survival, and we're very likely looking at an early hatch. Next slide, please. This is the forecast for precipitation, and we are actually looking at an increased probability of precipitation for areas of the prairies, southeastern Alberta and southwest Saskatchewan that could really use it right now. So I'm hoping that these probabilities are maintained, and those areas do receive some precipitation. And next slide, please. And you can see here, this is the snow water equivalent. So that precipitation is very likely to fall as rain. The forecast model is suggesting not very much happening in the way of snowpack for the spring. So of course that snowpack is very important for getting things going in the spring, and we're hoping the rains are quite timely when and where they do occur. Next slide, please. Of course, grasshoppers are food for a number of different critters, and I'm not going to go through the whole list because the list is very long. Just mention a couple of my favorites, or a few of my favorites. So Scaleo species egg parasitoids are tiny little wasps, so less than 1 millimeter long, that are attacking grasshopper eggs as they're laid in the soil. So they're actually going to deposit their egg into the grasshopper egg using their stinger or their ovipositor, squirt a little venom in there as well, and their larvae develop inside the developing grasshopper egg. The next one is tangle-vane fly, which has a really peculiar host finding strategy. So she is going to lay about 2,500 eggs on a fence post or another elevated structure, tree trunk, and those eggs are going to be distributed by the wind, and they're just going to randomly find their way to grasshopper nymphs. And I'm sure the females are scouting the area to look for the prevalence of grasshopper nymphs. They have, you know, great big beautiful eyes and excellent vision, so they're very likely scouting the area to hedge their bets on that. But it's still a very interesting strategy. And finally, Formica sanguinea, these are slave makers. So they actually attack the colonies of other Formica species, and they're going to take the developing pupae and larvae of the other ant species, and those ones become the lowest caste in their own colonies. So they take slaves. But these are also predaceous and aggressive. If you've ever encountered them, I have several colonies of these on my place, and really don't enjoy standing in the midst of their colonies because they're biters. And they will consume large numbers of grasshopper nymphs. Next slide, please. So migratory behavior, we certainly saw some of that with two-striped grasshopper last year. In the case of migratory grasshopper, where most of the work has been done, you're going to have a portion of the population that are pre-baked as short distance flyers, some are pre-baked as long distance flyers. So these are not learned behaviors, these are heritable traits. So, you know, a portion of the population is just comes pre-baked to do that. You can select for a greater number of long distance flyers when conditions become rough. So if there's resource shortage or semi-arid conditions, that is going to select for a larger number of long distance flyers. And when I say long distance, I mean long distance. I mean really long distance. So some work that was done with flight mills indicated that they can fly 10 hours or longer. And the reason it's 10 hours is they terminated the experiment at that point. There was no indication that the grasshoppers would stop flying. It's not like they looked tired or anything. They just said, 10 hours, holy cats, that's a lot. We should probably terminate that. So they do typically fly at flight speeds up to 20 kilometers an hour. So the possibility for them to fly 200 kilometers a day exists. They do tend to fly upwind. So that's going to limit their spread, but they can still fly great distances. So they can fly at night. Typically, however, they like to roost, and they can fly at great heights and have been a problem with fouling commercial aircraft windscreens. Not at heights of 30,000 feet, but you know, in the 10,000 feet area. Still pretty high. Next slide, please. So historically, this is from an outbreak in the late 30s. And this is just to illustrate the distances that can be covered. So the source of these populations was in the Dakotas. They covered on average about 100 kilometers a day and found themselves in Manitoba, southern Saskatchewan, southern Alberta, and all the way into Texas. And so why these mass migrations are potentially important is, if we go to the next slide, please. Of course, our neighbors to the south have also got some grasshopper issues. And you can see that there are some hotspots in the Dakotas and in Montana and in some other regions too. So we'll be reaching out to our colleagues south of the border just to see exactly what's happening with their grasshopper hatch and trying to gauge what our risk is going to be, in addition to trying to keep an eye on what's happening here. So next slide, please. Did develop some thresholds, or expanded the thresholds based on what are commonly forwarded in Western Canada. So different crops, different stages, and both the threshold in the field and in the ditch. So use again the data that's been accumulated in Western Canada and the thresholds that are forwarded here, as well as some information from our U.S. colleagues, both in Wyoming and in the Dakotas, and compiled those into this list. There's a lot to go through here, but you know, in short, greater tolerance for nymphs, they cause less damage. For cereals, 10 to 12 as a rule for large adults or for large nymphs and adults. Canola, soybean, more tolerant because they can take a lot of foliar damage without suffering a yield hit. Lentils and flour, flax and boll, not tolerant at all really for grasshoppers, 2 per square meter. And range and pasture, you're talking about a pretty good tolerance, at least for adults, and a very good tolerance for nymphs, but there is still a threshold there. And I've included some for the ditch. So I'll try to make this available through ministry website and the Western Forum website, Prairie Pest Monitoring Network as well. Next slide, please. So moving on to some other pests, we also do we have a contractor who conducts a survey for wheat midge, and this survey was conducted in 2023. As many of you are aware, the list of products available for control are now dimethoate, full stop, but there are a number of varietal blends with the SM1 gene that are still very effective. Next slide, please. So although we had a very dry year, although a bit of a wet spring with some very timely rains in some areas that contributed to upregulation of wheat midge populations. So you can see we still got some hot spots north of Regina and some minor hot spots in other parts of the province, but overall numbers are relatively low. You can also see that we concentrated our efforts in areas that are not suffering from protracted drought, but we need to move a little bit further north. So there will be a QR code for survey sign up, and I really encourage people request want everybody sign up for surveys so that we can get this information and provide more rigorous forecasts. So you never count out wheat midge. If you got timely rains, so that is more than 25 millimeters before the end of May, you can still see upregulation of midge populations. Next slide, please. We've also had some reports of Hessian fly near Cut Knife, Blaine Lake, Bigger, and Central Butte. The last report of these was in 2014. As a rule, they're regulated by at least 4 parasitoid species, and we did rear parasitoids out. This little eulophid, you can see that on the left, that was reared out at the Crop Protection Lab in Regina, and Tyler Wist reared another parasitoid out in his lab in Saskatchewan from different populations. So 2 generations of Hessian fly. The first generation can contribute to stunting and yellowing of wheat crops in the spring, and the second generation can contribute to broken stems, a little bit like wheat stem sawfly, although the rate tends to be higher. So we're keeping an eye on this one. I think it's a little premature to be terribly concerned, but it is one to definitely keep an eye on. If the parasitoids are not regulating it to the extent that they could be, this can be an economic issue and was an economic issue in some areas. Next slide, please. We had some problems with a true armyworm, also called armyworm cutworm. You can see the problem with common names. I think that's a really good example of it, but it is a generalist. It's a powerful flyer. It flies up from the United States. Of course, they're not flying as caterpillars. They tend to just plop as caterpillars. So it's going to be the adult moths that are flying, and they are generalists, but they've shown a real predilection for wheat and durum, and we saw some heavy populations near Outlook in Saskatoon. Next slide, please. We also saw some heavy populations of cereal leaf beetle, and you can see the characteristic damage on the right there, that stripping of the leaf. You can also see what looks like a little blob of turd, and that little blob of turd is actually a little blob of turd, but inside that blob of turd is that larva. So they produce what's called a fecal shield, and it's to protect them from parasitoids and from predators. So I don't know if that would work for people and lions, probably not. But it seems to be a decent strategy for them. However, they are really well regulated by a parasitoid. And you can see that larva without its fecal shield with the developing wasp larvae inside of it. So that animal is going to go into the soil for pupation and never to be seen again. The adult on the left there, all of these can cause economic damage. And we did have some reports of damaging populations, but wide occurrence of parasitism of these animals too. So any populations that are outbreaking should come under control by the parasitoids. Next slide, please. Wheat stem sawfly. This is the first year of an annual survey. And this is another one that is conducted by a contractor. 86% cutting at a site near Saskatchewan Crossing, above 70% cut at a site near Regina. Overall cutting and prevalence was relatively high. Sawfly stub was found in all but 2 fields. It is a native species that has crossed over to cereal crops and seems to do very well in them. We also had reports in RM438, and because this animal favors warm, dry conditions as well, we may be looking at an increase in occurrence of sawfly cutting. Next slide, please. And this is the actual survey. So the really high density regions occurred relatively further north than I was expecting. So we're going to actually be expanding the survey a little bit further north in the next year. Probably not with the same resolution, but you know, greater geographic area. But yeah, widespread high occurrence of these animals. Next slide, please. Flea beetles, not much of a flea beetle year this past year. We still had economic populations in the northeast, primarily with striped flea beetle Philotrita striolata, and some damage in in southern regions near Moose Jaw. Large numbers of overwintering crucifer flea beetle in the fall, but overall not really much of a flea beetle year. There's some indication based on modeling work published by Julie Soroka a few years ago that crucifer flea beetle is more drought tolerant than striped flea beetle. So the very dry conditions may be countering some of the differences in susceptibility to insecticides. It's not one thing; it's a bunch of things that are contributing to the species composition. So dry conditions seem to be trumping all though, and reducing the occurrence of striped flea beetle, at least in southern regions. Next slide, please. Cabbage seed pod weevil is invasive, continues to spread. Asterisks north and east. It does occur in Manitoba now, albeit at low levels. Adults can be economically damaging and contribute to bud blast, but larvae are really what are causing most of the damage through direct consumption of the seeds. So those eggs are going to be laid in the seed, one larva per pod. They cannibalize each other, so there can be only one. We had some limited reports of spraying for this animal, but not like we saw in a couple of years ago. If we could go to the next slide, please. Populations were relatively concentrated. So we actually saw a retraction from northern regions in 2022, and that was maintained in 2023. We saw some concentration of populations near Swift and Regina, but overall the geographic range was relatively lower than we've seen in previous years, although we still have the possibility of economic population. So this is definitely one to still monitor for. Next slide, please. Aster leafhopper. Alireza mentioned it was a pretty hot year for aster yellows, and aster yellows is vectored by this little animal. This is the aster leafhopper, and it feeds on a number of different plants, but the aster yellow seems to show itself the most in canola, or a lot in canola, and you can see the symptoms there. Large numbers of these insects were trapped, and of these infectivity was very high. So on average, about 16%. There was one site where infectivity was 68% of the aster leafhoppers that were captured. Dr. Wist has also developed a rapid lamp test in collaboration with colleagues at AFC. That can process samples in about 30 minutes to make a determination of aster yellow's occurrence or the phytoplasma occurrence. No thresholds associated with these ones. So products that are registered include dimethoate. Malathion is registered for use in canola, but not for leafhoppers. It is, however, effective for other leafhoppers in other crops. Next slide, please. Diamondback moth is one we continue to monitor for with in the past year a 63 trap sentinel system. You can see there was an ATV here that ran through some canola and picked up a few diamondback moth larvae. So they were numerous in some areas, although overall populations were low. We continue to monitor these into August to look for local upregulation of populations. So early arrival, they're going to speed through a number of generations. Of course, you get an increase in numbers with each generation. So we're looking for local flare-ups in addition to time of arrival to get an estimate of what we're looking at for populations. We did see some spraying in July and August, and unlike 2021, where we had some cases of possible group 3 insecticide resistance in these animals, we did not see any of that in this past year. North Dakota actually issued a statement in 2021 to not use group 3 insecticides. So that is, your Desis and cypermethrin and Matador silencer on these animals because of widespread group 3 resistance. They move up from Florida, they fly up from Florida and northern Texas, northern Mexico, where they're feeding on brassicaceous veggie crops. And they're hit frequently with insecticides, and resistance to a broad assortment of insecticidal groups has been documented. There seems to be a winnowing effect, on their migration up from those parts of the world where they are until they arrive here. So those that are resistant don't seem to survive that flight very well. So exactly what's happening, uncertain, but you now know what I know. Next slide, please. So yeah, this is the Diamondbacks survey. So certainly, some relatively early arrival, and with the warm temperatures, increase in numbers. These animals don't overwinter here, or they do at such low levels that it's inconsequential. So don't consider this to be a risk for next year or this coming season. This is what happened last year. Next slide, please. Bertha armyworm is another one that we continue to trap for, 301 traps, and sincerely thank you to everyone who participates in our trapping programs. This one is a cyclical outbreaker. Its populations are usually controlled by pathogens and other natural enemies, and we're really kind of overdue for an outbreak of these animals. We will continue to monitor for them, but we're at a real low ebb. So the last minor outbreak, 2016, outbreak coming soon. We don't know. We will continue to monitor, but the timing is certainly right for one. Next slide, please. So yeah, the Bertha survey, really not much to write home about here. And no reports of economic issues with these. We did have some reports in the Northeast where there was some thinking it might have been Bertha. This turned out to be clover cutworm. Some reports in other regions which turned out to be some other climbing cutworms and loopers. So no confirmed economic populations of Bertha this past year. Next slide, please. Cutworms, I mentioned before, some significant issues with some climbing cutworms, especially clover cutworm and canola, and some other issues with alfalfa looper and cabbage looper in some other regions as well. For subterranean, mostly redback, some pale western cutworm as well. Some reports of spraying. Both of these species, both pale western and redback cutworm, like it dry. And areas with broadleaf weeds are really good areas for the females to lay their eggs. They're not very host specific, so they're really looking at a broad assemblage of different weed species to lay their eggs. Next slide, please. Lygus, the nominal threshold, as I'm sure most of you are aware by now, has been altered to 2 to 3 per sweep, late flower to early pod, full stop. So that's at all control costs, all commodity prices. We had few reports of spraying in canola in 2023. Where we did see some action though was in yellow mustard, and we don't currently have a threshold in yellow mustard. The lit review that I did and some others have done looked in the lit as well. Does suggest that yellow mustard is pretty tolerant of Lygus feeding, although we did have some reports of some heavy populations near the U.S. border in the southwest. Faba, however, is very sensitive to Lygus feeding, and flowering to mid-pod, most vulnerable, and you can see the piercing, sucking mouth parts. They actually inject a salivary enzyme called polygalacturonase, and they need to liquefy their food to take it up, and that's what this digestive enzyme does. But it causes a lot of quality issues for Faba, which can be a real problem for marketing. So Sean Prager's lab and his master's student Teresa she is just winding up and have been looking at effects of Lygus on Faba, and look forward to them finalizing the results. Next slide, please. I'll present This is a map that was developed by Teresa Cordero, just looking at different Lygus species present in FABA and the relative numbers and the species present. And so look forward to that final report. Next slide, please. Pea leaf weevil. This is one of the last insects we'll talk about. This is another invasive, and the numbers of this animal were very low for a number of years, to the point where there was some open discussion of whether or not this was still even really a pest. And of course, they overheard us talking like that, and you'll see the map. So the adults can be problematic. You can see the notching on the leaves, which is quite characteristic. Yield effects, however, are almost always associated with larval feeding. And so they're laying their eggs, they're just dropping them on the surface of the soil. She's just trundling across a field and just laying eggs and dropping eggs at random. She'll live for about 2 to 3 months and will continue to lay eggs that entire time, not in clutches, but individually. So these eggs are going to hatch up. The larvae are going to find the nodules, rhizobium nodules, tunnel in and munch on the contents of those nodules. And that can result in an almost 30% yield hit. So they can be a substantial pest. And we did see some population increase. And we can go to the next slide, please. So I don't know if many of you remember from previous years, we were looking at fields of green. And we saw a real upregulation in 2022, expanded our survey a little bit further north, but you can see a real upregulation of populations there. So the best solution for these animals is insecticidal seed treatment. There are products available with thiamethoxam, the neonicotinoid, which are effective. Foliar application for these animals. There isn't really hard data to support its efficacy. So if you're in a region with a high risk, my recommendation would be to look at seed dressing. Pardon me. Next slide, please. And this is the last insect. So spotted winged Drosophila. This is a survey that's led by Forrest Sharp and includes members of our regional specialist group, as do a number of these other surveys and our SARM plant health technical advisors, in addition to a number of other people. And my sincere thanks go out to everyone who participates in these, and to Carter Peru, who does an excellent job of cat herding, of keeping everybody pointing in the same direction. So hats off to everyone. With that, spotted winged Drosophila, for those of you not familiar, is considered one of the major invasive pest species on the planet right now. There was some indication that it wouldn't overwinter in Saskatchewan because of the cold winters. Forest surveys have indicated, using his sentry traps and other lures, that they are very numerous in the spring, which is strong evidence that they are overwintering very happily in this part of the world. They will feed on a variety of fruit crops. They will feed on tomatoes. They will feed on, if it ends in berry, it is a host plant. And you can see that developing larvae inside that developing fruit, and that ruins that for commercialization. So sentry traps and lures have become the standard for this survey, and are equally attractive to both males and females. And if we go to the next slide, bit of a curious puzzle happening here. So on the left are females that are captured, and on the right are males. And for some reason, there were no males that showed up till relatively late in the season. And what the heck is going on here? Because they're not parthenogenetic. They don't lay unfertilized eggs. Mating is required for the production of the next generation. So where are the males? Do not know. At the pub, I suppose. But yeah, no, really, it's a real head scratcher. And we will continue to monitor this. And finally, next slide, please. We've begun a survey for Richardson's ground squirrel. This is an online grower survey. We had 174 responses or sites from across the province, and we strongly encourage everyone to participate in this. We're going to do it again next year, and of course, more data is better data. So really appreciate everyone who signed up. It's about a 5-minute survey. Really appreciate everybody who signed up. So questions included acres affected in a variety of different settings, damage severity in a variety of different settings, control techniques and satisfaction with that control. We're looking at this data currently and lining this up with some weather data to see if we can't develop some predictive power for Richardson's ground squirrel in upcoming years. And just to take a quick look at the data for the next slide, please. These are the total acres affected per person reporting. So yeah, I think for the questionnaire, this could be tailored a little bit more specifically, but it does give an idea of the breadth and width and severity of some of the hotspots last year, and does suggest that with continued warm, dry conditions, which are putatively associated with increases in population, that we're going to continue to look at pressures associated with these animals. And I think my final slide. I did mention before a QR code, and as promised, there it is. Please point your phone at it and sign up to allow us access so that we can continue to conduct this the survey work. And with that, I will take any questions. And thank you for your patience with audio issues.
Yeah, thanks, James. Yeah, another reminder, I guess if you have a question, just throw it in the chat box. We've got a few questions here for you already, James. How are you expecting the lower than normal snowfall this year affecting insects in 2024?
Yeah, it depends on insects. So I mean, in the case of grasshopper, it has the because of that lack of a protective effect of the snow, it has the potential to maybe put a little dent in the grasshopper egg survival. Is it going to be major? Probably not. I don't think. I think pests or animals like wheat midge that are really dependent on adequate soil moisture to stimulate them coming out of diapause. I think that lack of moisture could negatively impact them, and they could enter into a delayed diapause and just hedge their bet till next year in regions with low snowpack and low rain. But in regions with 25 millimeters of rain before the end of May, that trumps all. So as long as they get adequate moisture, then they're going to be stimulated to complete their development. Yeah, it's a bit of a mixed bag for a number of different insects, because a lot of them have different moisture requirements. Some do better under moist conditions, some do better under dry conditions. I think the reduced snowpack contributing to dry conditions favors grasshoppers, favors wheat stem sawfly. Those are the two primary ones that I think will be of concern. Its effects on crucifer flea beetle uncertain. It will probably have a negative impact on striped flea beetle.
Okay, next question. Are there natural ways to control grasshoppers / grasshopper eggs that can be applied to a field that is not chemical?
Well, yeah, that's an interesting question, and there are some, we'll say, mixed results on that. So there's some may remember hearing mention of the Rocky Mountain locust, and if you haven't heard of it, don't be embarrassed, because it's been extinct since the late 1800s, and that used to be our primary pest on the Great Plains of North America. And it's thought one of the factors that contributed to its extinction. So going from a major pest contributing to famine continent-wide to extinction was tillage. That said, that's a different species than the ones we're dealing with, and the environment was considerably different. It was a much more natural system at that time. There were a lot of other things that were happening. Widespread movement of populations, First Nations populations, the movement of Europeans into the West, the introduction of agriculture in general, or intensive agriculture in general. But it's thought that tillage may have contributed to this. There's evidence that shallow tillage, because it limits the numbers of potential host plants, weeds, primarily weeds in a field, can reduce the predilection of these animals, the species that we're dealing with now, to invade fields, and because the first instar nymphs don't move very far, that can actually contribute to their starvation. So that's a possibility. Deep tillage, however, It doesn't seem to be very efficacious. It just sort of, you turn the soil in big clods, and the grasshoppers just seem to emerge from that anyway. But the jury is really still out even on shallow tillage. I mean, there's as many examples of it being effective as not.
Okay.
Other factors that can be influential as well is your choice of crops. I mean, we're dealing primarily with two-stripe grasshopper, and work by Owen Olfert and some recent work that we've done in collaboration with Ag Canada has indicated that field peas, pardon me, are a non-preferred host plant. So two-stripe grasshopper really doesn't like field pea very much. There's some evidence that oats is a less preferred host plant as well. And there's some evidence that there's some differences in wheat varieties in their susceptibility to grasshoppers. We haven't seen it so far in our work. But there was a former commercially available variety called HY320, no longer available, that was demonstrating some resistance to two-stripe grasshopper. So we've incorporated that into our tests, and we'll see how it shakes out. But so far we haven't seen differences in the susceptibilities of the different wheat varieties.
Okay. The code on the screen for the pest survey sign up doesn't seem to be working, but you can find information in the sign up in the handouts located on the side. The QR code for the CEUs will be shared after the question period. So next question here, James, is how effective is dimethoate to wheat midge?
Conditionally vary. It doesn't have the ovicidal effect or the larvicidal effect that chlorpyrifos did. So Lorisban would volatilize, and you get this vapor effect that would just kind of permeate all the nooks and crannies. Dimethoate doesn't really do that. So you're reliant, although it does have systemic activity. You're almost entirely reliant on getting your timing right. So you need to hit the adults as they're emerging, and if you can do that, it can be a very effective product.
Okay, this is just a comment from a participant. They said that they saw sawfly infestations in the Watrous/Young area in 2023. When do aster leafhoppers actually transfer the aster yellow's disease, or what is the timeline from the infection to symptom development in canola?
I'm going to defer to Alireza as far as infection to symptom development, but as far as infectivity, it's when they start feeding. So pretty much the moment that they arrive, when they fly in or are pushed in on the high altitude winds, they're going to start feeding on plants. And of course, canola isn't their only potential host plant. They will feed on a broad assortment of different host plants, and there has been some work at U of S on their host preferences and suitability of particular host plants. And I'm getting a little off in the weeds here, but it seems like canola actually isn't a very good host for the insect, although it seems to be a pretty darn good host for the phytoplasma. So yeah, infection is going to happen the moment feeding begins.
Okay. Are soybean weevils being monitored? I saw high populations on the Saskatchewan-Manitoba border this year.
Soybean weevils or soybean aphid?
It says soybean weevils.
No.
Okay. How is the ground squirrels, or how is it that ground squirrels affect the crops?
Oh yeah, in a number of different ways. Yeah, they can be a real pain, either through direct consumption of crops, and they can cause a lot of damage to specific crops. I mean, many of you, I'm sure, are familiar to seeing, 20, 30 acres turn into a lunar landscape through direct consumption. Their burrowing, of course, can be problematic for equipment. Can damage equipment. And can be a real problem for livestock as well. I think an important consideration too, and where I encourage some tolerance and the use of a threshold for Richardson's ground squirrel is they are really important for ecosystem functioning. And so to be somewhat tolerant of them, obviously if the population's out of hand, then you need to do something about it because they can be a serious economic pest. But tolerance of them and tolerance of their predators, I think are really important and will contribute to a healthier environment, less prone to outbreaks.
Okay, that was the last question there. So at this time, we would like to thank everybody for taking the time to join us for the event.